Large reference databases of molecular responses to systematic perturbations, such as the connectivity map, are invaluable resources for network-based drug discovery. Connectivity map comes with its own set of challenges and limitations. It's heterogeneous and multidimensional. Related methods usually focus on a single disease context, and it has not been sufficiently integrated with cheminformatic data. Here we present a computational framework that uses links CMAP to build cell type specific gene perturbation networks and integrates these networks with an intermediary panel of disease phenotypes and cheminformatic data to jointly rank cell lines and perturbations. To address the first challenge, we introduced the quantile-based instance z-score consensus method, or QuizZ. Briefly, for each gene, we assign a perturbation z-score to each perturbation instance against the background of all perturbation instances on that gene. We require that there be a sufficient number of differential instances of a perturbation for it to be deemed significantly affecting a gene. To determine this number, we define a flexible consensus threshold based on the z-score distributions. Perturbations that pass this flexible consensus threshold are considered differentially affecting the expression of that gene. Repeating this procedure for all genes and perturbations in every cell line and aggregating all the differential gene perturbation links, we build global cell type specific gene perturbation networks. QuizC networks include the majority of the small molecule perturbations tested on the CMAP cell lines, with 34 out of 60 cell type specific networks having over 80% of the perturbations tested in each respective cell line. We observed that the largest hub perturbations, meaning perturbations modulating the expression of a large number of genes, are generally cell type specific and are not shared by many cell lines, whereas the perturbations with fewer targets can be present in a large number of cell lines. On the other hand, genes targeted by many drugs tend to do so in a large number of cell lines and vice versa. In terms of individual gene perturbation interactions, we see little overlap between pairs of cell types. To supplement our findings on the structural uniqueness of QuizC networks, we quantify the degree of information redundancy or structural reducibility in multilayer networks. Within this framework, QuizC networks are highly non-redundant with a reducibility metric of 0.16. In terms of what we call topological drug specificity, which is defined as the inverse of the number of cell lines a drug appears in times the mean out degree of the drug, QuizC networks are more specific than two other links CMAP-based methods, the moderated z-score mod-z and the characteristic direction CD. 48% of QuizC cell type specific networks have a significant enrichment of known drug target interactions. In terms of clinical development, we find a balanced representation of experimental drugs, investigational drugs, and approved drugs. Investigational drugs are present in more cell lines than experimental drugs, which in turn are present in more cell lines than approved drugs. In terms of out degree, investigational drugs have a higher number of target genes compared to approved and experimental drugs. To address the second and third challenges mentioned at the beginning, we first defined the signature congruity score, SCS, to quantify the agreement between two gene signatures in terms of their direction of effect. Extending the SCS calculation to all diseases in a comprehensive disease signature panel comprised of more than 500 diseases, we define pathophenotypic congruity profiles, or PCPs, which are vectors composed of SCS values. We calculate PCPs for each perturbation in each cell type specific CMAP-derived network, as well as for the input signature. Finally, we introduce the pathophenotypic congruity score, PACOS, which quantifies the similarity or dissimilarity between the PCP values of any input gene and cell line perturbation pair. We rank each perturbation and calculate ROC curves for each line, and also perform the same operations on shuffled ranks to come up with a ranked list of cell lines. The integration of PACOS and tool score significantly surpasses either approach alone in all three cases, with QuizC outperforming both the moderated z-score and characteristic direction, both forward and reverse mode. Due to its being agnostic to the specific input signature, tool score by itself performs poorly compared to PACOS and similar to random expectation. In addition to this internal benchmark, we perform an external benchmark with another linked CMAP-based drug prioritization method, L1000-CDS squared. L1000-CDS squared performs better than tool score only, while it does comparably with PACOs. Combined with tool scores, all three PACOs versions significantly surpass L1000-CDS squared, with QuizC having the highest improvement. As a proof of concept, we use as input the set of differentially expressed genes in SARS-CoV-2. Running the PACOs tool nested prioritization in reverse mode, we discover 4 cell lines that have significant areas under the ROC curve values. Among those, the compound with the highest PACOs score is SP203580, which is a potent and selective p38 MAP kinase inhibitor. As in evidence of direct relevance to COVID-19, its antiviral activity has recently been reported. It has 2 edges in the respective QuizC network connecting to IER3 and FGF1, which are both implicated in MAP kinase signaling and are related to stress response, inflammation, and tumorigenesis. Looking at the PACOs intermediary disease phenotypes, we see a host of neoplastic, inflammatory, and neurological diseases for which the same compound has been studied, which may suggest potential common mechanisms of action. In conclusion, our analytical framework surpasses currently available methods in terms of predictive performance and offers a feasible blueprint for a cell type specific drug discovery and repositioning platform that accounts for multiple disease phenotypes. With this, I'd like to thank my colleagues at Brigham and Women's Hospital and thank you for your attention.